So for lab number 2, we're going to be getting more familiar with the yeasts, and we'll be looking at 3 different yeast organisms: Candida albicans, Saccharomyces, and Geotrichum. The first thing that we're going to do is streak these from a stock culture onto a cornmeal agar plate. This is a specialized media that's going to allow the formation of germ tubes if it's a Candida organism. So you'll notice that Kim is using a loop rather than the probe that we used before, and she's going to pick it up just like it was bacteria, and she'll be streaking it for isolation on that plate just like you would do with bacteria. Each of those streak marks will come back later and take a plug out of the agar after it's grown, and we'll look at that under the scope to see if we're seeing pseudohyphae. Now with these yeasts, you also can make a cotton blue wet mount, just like you did with the true fungi, and you'll be taking a portion of that culture and plating it on a slide, or placing it on a slide in the cotton blue, and then you'll put a cover slip on that, and you'll be able to come back later and look at it. So Kim's using a wooden stick this time to pick up the inoculum. Works a little better for this rather than streaking the plate with it. It's better to use a loop because you can flame the loop. And then you'll place a cover slip on there. Now you'll have still images of these different yeast organisms that will allow you to differentiate Geotrichum from Saccharomyces and also from Candida. Okay, so we're coming back 24 hours later. You've allowed the yeast to grow on the plate, and we're going to actually cut a little plug, a square or rectangular plug, out of the agar, and then we'll take that and place it on a slide and put a cover slip on top of that, and that will enable you to see the growth and the pseudohyphae if it is Candida. The other organisms will not produce the pseudohyphae. So when we place the cover slip on that, we're not going to squash it down. We're going to just place it lightly on top so it sits up on top of the agar, and then the hyphae won't be disrupted. You'll be able to see them growing, but they won't be disrupted. So the last test you'll be doing with the yeast will be to look for germ tube formation in rabbit plasma. You'll perform this test by inoculating a small tube of rabbit plasma with some of your yeast culture, and that can be done using a sterile stick. So that tube will then be placed in the incubator and incubated for maybe an hour, and then you'll come back and you'll take a small sample of that, maybe 1 drop, and place it on a slide. Then you'll place a cover slip on that. And you'll look at it under the microscope.